Sorry for the delay in tonight's webinar. We were experiencing some technical difficulties, but I want to wish you a good evening and welcome you to the Cure HHT webinar series. I am Nicole Schaefer, Director of Professional and Medical Programs for Cure HHT. Thank you for joining tonight's webinar, The Science Behind the 2019 Scientific Conference. Just a few reminders, tonight's webinar is being recorded and will be posted in the News and Resource section of our website. After the webinar, you will receive an email with a short survey. Please take a few minutes to give us your feedback. Your input is critical to the development of future webinars. To help reduce background noise, everyone's phones or microphones are muted. You can ask questions throughout the webinar by simply typing your question into the gray panel on your screen. I will ask as many questions as time allows during the second half of the webinar. Please remember to ask questions related to today's topic so the answers benefit everyone on the webinar. And the executive summary of the 2019 Scientific Conference, along with all of the abstracts, have been published in the professional journal Angiogenesis, and these articles are in the Cure HHT resource library. I will provide a link in the email you'll receive after the webinar. But before we begin, I'd like to introduce our first speaker, Dr. Philippe Marambaud. Dr. Marambaud is currently Professor and Director of the Laboratory of Memory Disorders and Vascular Biology at the Feinstein Institute, Northwell Health in New York. He obtained his PhD in biology in 1998 from the University of Nice, Sophia Antipolis in France. He then completed a postdoctoral fellowship in 2000 at the Icahn School of Medicine at Mount Sinai, New York. He was recruited as faculty at the Albert Einstein College of Medicine in Bronx, New York in 2004, and as laboratory head at the Feinstein Institute, Manassas, New York in 2005. Since 1995, Dr. Marambaud's research has focused on the molecular basis of neuronal degeneration in Alzheimer's disease and other dementias. In 2015, his laboratory significantly expanded its activities to include programs aimed at studying HHT. In cell and mice models for this disease, he seeks to increase our understanding of the molecular basis of HHT, but also to identify disease-modifying interventions. Dr. Marambaud, I now pass the program over to you.
Good evening, everyone. Thank you, Nicole, for the kind introduction, and thank you to all the Cure HHT staff for organizing this exciting webinar tonight. So my job today will be to summarize the most significant data that were presented at the last HHT International Scientific Conference. And before starting, I would like to say that this is in any way an exhaustive list of all the presentation, and I apologize in advance if some work is not cited. So we heard a lot of interesting new data on the mechanism of mechanotransduction in endothelial cells last year. The interest has grown significantly in the past few years for a role of these mechanisms and for the effect of blood flow more generally in the context of HHT pathogenesis. Two of our invited speakers presented data on this topic. So Holger Gerhard from the MDC in Berlin first presented groundbreaking cell signaling and high-resolution imaging data on vascular remodeling during angiogenesis, lumen formation, and vascular patterning. Data were presented that explain how endothelial cells sense the blood flow via the primary cilia to control BMP signaling and to prevent excessive vascular regression. He elaborated on how endothelial cells migrate from veins to arteries, mechanisms that could have direct relevance for AVM formation in HHT. Elite Sima from Oxford University in UK also gave a great overview of the role of the PCAM1 V-cadherin VEGF receptor 2 complex in mechanotransduction and blood flow sensing in endothelial cells. We also learned about a new role played by plexin D1 as an upstream sensor of shear stress and blood flow. During the endoglin ALK1 signaling session, several talks addressed the important question of whether BMP10 is a physiological ligand for ALK1 and whether its deficiency could lead to AVM. So new data in endothelial specific inducible BMP9 and BMP10 knockout mice were presented by Dr. Choi from Paul Ho's lab. The team generated data showing that BMP10 knockout mice, but not BMP9 knockout mice, develop anemia and GI bleeding, and that BMP9 plus 10 double knockout mice have a more severe phenotype. Postnatal retinal AVMs were detected in the BMP10 knockout mice, but not in BMP9 knockout mice. So the team proposed that BMP10 is the most relevant physiological ligand of endothelial endoglin ALK1 signaling and thus has therapeutic potential in HHT. A similar question was addressed in zebrafish models, which relates to the unique and overlapping functions of BMP9 and BMP10. This work was presented by Teresa Capasso from Beth Roman lab from UPenn. We learned that BMP10 plus BMP10-like double mutants resemble ALK1 mutants by forming embryonic cerebral AVMs. So this model also developed skin, liver, and heart vascular pathology during adulthood, which is not the case for BMP9 mutants. So low systemic vascular resistance and high output heart failure was also reported in the BMP10 mutant fish. The team concluded that BMP10 is a sole required ligand in endothelial cells in the adult fish. So the debated question of whether BMP9 and BMP10 form homodimers and heterodimers was also addressed in a presentation by Andy Hicks from Penn University. He presented data that pro-BMP9 and 10 are produced as a mixture of monomers and dimers, and that BMP10 monomers are 40-fold less potent than their corresponding dimers. We learned also that a BMP9 plus 10 heterodimer can form both pre and post secretion from the cells, data that confirm Sabine Bailey's data of the presence of a heterodimer BMP9 plus 10. Exciting new evidence was also presented showing that endoglin and ALK1 form a linear signaling pathway involved in proper AV network development during angiogenesis. These data were presented by Yong Kim from Paul Ho's lab. The team used inducible ALK1 or endoglin transgene overexpression system in endoglin and ALK1 inducible knockout mice to analyze the effect on AVM in the skin wound model and the developing retina, two models that are routinely tested in the lab. ALK1 overexpression, but not endoglin overexpression, prevented skin and retinal AVMs in endoglin and ALK1 inducible knockout mice, and BMP10 infusion, but not BMP9 infusion, could inhibit AVM in endoglin inducible knockout retinas. So these data are very important because they suggest that ALK1 overexpression or its activation via, for instance, BMP10 treatments can be of therapeutic value in HHT. New data on the mechanism that links BMP9 with liver vascular malformation were presented by Sabine Bailey from the CEA in Grenoble. We learned that BMP9 deficiency in mice in a 129 genetic background develop liver sinusoidal fibrosis starting at 15 weeks of age. Inflammation appears at the same time, while sinusoidal vessel dilation develop later. The team concluded that BMP9 plays a role in liver homeostasis, liver sinusoidal endothelial cells fenestration, and protects against perivascular fibrosis. During the genetics of HHT session, groundbreaking data were reported by Daniel Snelling from Duke Marshuk's lab from Duke University, which support the model of biallelic somatic mutation in endoglin and ALK1 as a trigger for focal clinical manifestation in HHT. Somatic mutations were identified by deep sequencing in 9 out of 23 lesions, 5 in endoglin and 4 in ALK1. Evidence was obtained that these somatic mutations occur at a frequency of 0.5 to 9% and only in the Y-type allele of the constitutively mutated gene. So unique somatic mutations were found on different lesions from the same patient, suggesting that they are seeded by unique mutation events. So this is the very first evidence that biallelic loss of endoglin or ALK1 can trigger lesion development in HHT. So this work has been preprinted in bioRxiv right after the HHT meeting and is now peer-reviewed and published. In the same genetic session, exciting data were also reported on the involvement of new causing mutations in non-coding genetic regions that lead to splicing changes. So this work was presented by Whitney Wuderschak-Donahue. The team used next generation sequencing to identify several intronic hotspots. Inclusion of these regions in genetic testing might improve HHT diagnostic sensitivity. So, invited speaker Dennis Adams presented a great overview of the role played by ERK and PI3K-mTOR signaling in vascular malformation diseases, and how selective pharmaceutical targeting of these two pathways using drug repurposing strategy has improved treatment of this disease. One of these drugs is sirolimus, which has produced spectacular results in some forms of vascular malformation diseases. We also heard data from Santiago Ruiz from my lab that in the neonatal retinal model, sirolimus has greater efficacy in reducing AVM compared to its macrolide analog tacrolimus. We propose that sirolimus works by inhibiting mTOR and rescuing SMAD158 signaling. In vitro evidence suggests that sirolimus works by activating ALK2 in endothelial cells. So this work is now published. We have also reported data indicating that combination of sirolimus with a RTK inhibitor nintedanib could provide additional beneficial effect by further reducing AVM formation in this HHT mouse models. Finally, interesting evidence was presented by Luisa Maria Botella that the drug etamsilate has anti-angiogenic effects both in vitro and in vivo in HHT patients by reducing epistaxis incidence. So, in addition, Johanna Frozen, on behalf of invited speaker Ivan Radovanovic, provided a great overview of the genetic origin and genetic predisposition leading to brain AVMs. He elaborated on the recent data showing that gain-of-function somatic mutations can induce lesion development in VM diseases, but also in sporadic brain AVM diseases. Landmark papers reporting KRAS and MAP2K1 somatic mutation in brain AVMs were presented, and he discussed the role played by two signaling pathways, PI3K, AKT-mTOR, and MAP kinase in brain AVM pathogenesis. A mechanism that could lead to therapeutic opportunities and an approach that was also discussed in great details by Dennis Adams for VM diseases. We also heard interesting data in mouse models from Sonali Shaligram from Hua Su's lab at UCSF, demonstrating that clonal endothelial cell expansion of ALK1 mutant cells, which are basically ALK1 knockout cells, is occurring in a brain AVM model. A brain and EC-specific KRAS G12D inducible transgenic model was also presented by Joshua Wythe from Baylor University. We learned that the mice develop vascular dysplasia and AVMs in the brain. Shunts in a zebrafish models were also detected, and MEK inhibition reverses KRAS-driven AVM and excessive sprouting in this fish model. We also learned that MR angiography with gradient echo and flow compensation techniques could be used to detect brain AVM in ALK1 and endocrine inducible knockout mouse models that were developed in Paul Ho's lab. Impressive longitudinal studies were performed, and lesion formation was followed over time. We heard new evidence that increase of VEGF level in the brain of an ALK1 inducible KO model increased brain AVMs and mouse mortality, and this was presented by Hua Su from UCSF. We also heard exciting new data in a non-endothelial specific SMAD4 inducible knockout mouse model from Strider Meadows from Tulane University. The team reported that RNA sequencing and chip sequencing analysis prioritized about 200 genes that are deregulated in the retina of this new SMAD4 knockout model. Angiopoietin 2 was found to be elevated, and its immunoblocking using a specific anti-angiopoietin 2 antibody reduced in a spectacular manner retinal vascular pathology, including AVMs. The blocking antibody not only prevented, but also reversed vascular pathology. So this work is also published now in the journal Circulation. Exciting new data from Ellen Arthur from Newcastle University in England in adult endothelial cell-specific endoglin inducible knockout mice were reported, showing high output heart failure consecutive to peripheral AV shutting. Heart hypertrophy, aortic pressure, and cardiac pressure were reported, increase in cardiac pressure were reported in this model. Brain shunts and extensive AVM in the pelvic region were also identified. High local VEGF expression in the pelvic region was proposed to be a key driver for vascular pathology development, and the use of an anti-VEGF receptor 2 antibody reduced pelvic AVMs and heart pathology in this model. Important new data were also presented by Wei Li from Cambridge University in UK on the role of surface and soluble endoglin in BMP9 signaling. We learned that secreted endoglin purified from human blood is mainly monomeric, and that secreted endoglin binding to pro-BMP9 releases mature BMP9 and remains bound to secreted endoglin. Importantly, Wei showed that data were reported showed that secreted endoglin is not a ligand trap for BMP9, meaning that it does not prevent BMP9, but in the contrary, might help to recruit and process pro-BMP9. The team also showed that cell surface endoglin is required for secreted endoglin BMP9 complex signaling. And again, this work is also published and in the Journal of PNAS. We also learned from work reported by Thomas Paulinus from Frank Lebrun lab that endoglin level should go below a certain level, certain threshold to induce disease development. They showed that endoglin expression varies to support vascular function, and haploinsufficiency may vary in effects depending on the basal level of endoglin. The team also reported that endoglin levels are lower in HHT-affected tissues, such as the liver, lungs, and skin. Thus, organ susceptibility might be driven in some way by endogenous endoglin expression. One of our invited speakers, Christoph Gibson from Recursions Pharma, presented new approaches using automated biological platform, machine learning, and software-assisted classification, and an identification of drugs for disease treatment. He presented new data on a drug called REC 65075, which demonstrated good efficacy in reducing anemia and GI bleeding in an EC-specific inducible ALK1 knockout model. During the cell biology session, we also learned that impressive progress were made in the development of a microphysiological disease model of HHT. This was reported by Chris Hughes' lab. This is a microfluidic vascularized microorgan system formed by primary human endothelial cells co-seeded with a stromal cell. We learned that the system self-organizes in vitro into perfused blood vessels that can form aberrant vessel networks upon ALK1 silencing in endothelial cells. A liver and brain system was presented. A possible exciting application for this in vitro system is, of course, drug screening. And to conclude, invited speaker Mark Kahn from UPenn presented new data on the role of the gut barrier and TLR4 signaling in the pathogenesis of CCM, cerebral cavernous malformation. We learned that glucocorticoid receptor signaling in both the epithelial and endothelial cells is required for this mechanism. Thank you very much for your attention, and I'll take any question that you might have at the end of this session. Back to you, Nicole.
Great, thank you, Dr. Marambaud. Obviously, basic science is probably over most of our heads, but we do have a lot of questions that came up during your presentation that ties it back to how, you know, it will advance treatments, what this means to the HHT community. So people were definitely energized and excited by what you had to say. So I'm going to now switch over the presentation to Dr. Andy White. And while he sets up his computer, I'm going to introduce him. Dr. White is the James P. Keaton Professor of Pediatrics at St. Louis Children's Hospital. He's a graduate of Brandeis University. He earned his master's from the University of Chicago prior to completing his MD degree at the University of Texas Southwestern in Dallas. He had a residency training in pediatrics at St. Louis Children's Hospital, which was followed by a fellowship training in immunology and rheumatology at Washington University prior to joining the faculty in 2000. He has been the Pediatric Residency Program Director since 2002, and in 2017 was named Vice Chairman of Education in the Department of Pediatrics. He served as the Division Chief of Pediatric Rheumatology for 13 years and has been the co-director of the HHT Center of Excellence since 2006. Dr. White has won the Distinguished Service Teacher of the Year Award 5 times and was an inaugural Philip R. Dodge Scholar of Pediatrics. In 2011, he was recognized with the Distinguished Clinician Award from Washington University. And now Dr. White will take us through the clinical science summaries from the 2019 HHT International Scientific Conference. Dr. White?
Thank you, Nicole. And I guess that wraps up our allotted time. Thanks for that long introduction. I'm sorry. But that's all right. I'm happy to be here. It's very exciting. And thanks for Philippe for giving a great talk. I have to say, as a clinician at this conference, I was more intrigued and interested in the basic science talks because I think a lot of progress is being made there. And I really look forward to how things will play out in the HHT field in the next several years. Great. All right, I can't hide that. Let me move this over to the side. Fantastic. Okay, can everybody see my slides? Okay, fantastic. Can I move my slides to the next slide? That's the question. There it is. Great. Okay, so thanks to everybody for putting together a fantastic conference, and to, of course, the HHT Foundation, the Cure HHT Foundation, and to Puerto Rico for hosting us, and for all the physicians and patients and interested parties who were able to make the trip. It was a great event. I'm going to talk a little bit about some of the what I perceived as the highlights from the clinical sessions. And there, as Philippe pointed out, there is no way we can talk about everything that was discussed. So these are just some highlights from several talks in each session. And I break mine up by sessions, and you can look at these in the abstracts that will go out later. So session 1 for me started things off on a great foot. So Dr. Oliger talked about he actually gave a very nice sort of cogent explanation of vascular lesions in the liver. And it was so clear that I was able to understand the anatomy better than I think I ever have before. He talked about the importance of identifying some complications in the liver, such as cholangitis and chronic liver injury, that I think we don't necessarily think about as much in HHT. He talked about some newer modalities for imaging and some important things that help us distinguish between various types of liver masses such as focal nodular hyperplasia, which is a benign mass that's relatively common in HHT, from nodular regenerative hyperplasia and cholangiocarcinoma, and even hepatocellular carcinoma, things that are not benign and need to be dealt with. He discussed phase contrast flow MRI and 4D flow, which are newer tools that probably will have some very useful clinical applications, particularly in response to therapy. Take-home message, liver MRI is a new direction for assessment, and we will probably be getting more liver MRIs in the future. Another highlight from session one for me was some of the diagnostic tools in HHT. I think we are all familiar with the Kurosawa criteria, but there were other areas that came up that were interesting. So, for example, Kevin White had noticed that some patients seem to have elevated ammonia levels, particularly those patients with ALK1 mutations. And this puts people at risk for hepatic encephalopathy. And he is now beginning to actually screen patients with ammonia levels to head off any complications from that ahead of time. Lawrence Young talked about subaortic membrane. So this is a problem, like a little fiber that forms below the aorta that can be seen on echo in patients with HHT. And it's a sign of turbulent flow, and it may be an indicator of high output heart failure, and it does seem to correlate with a poor prognosis. So pay attention was the lesson to us clinicians for subaortic membranes. Another talk from Vanden Heuvel was about comparing MR versus CT to detect pulmonary vascular malformations. And it turned out that MRI did miss a few, so CT was more sensitive. They missed about 16%, but only missed 2% of the sort of more clinically significant ones. This is an important step forward in what we hope in the future will be our goal of reducing radiation involved studies such as CT scans. So promising work on pulmonary MRIs. And then regarding echocardiograms and the amount of shunting that happens, there's a technique called grayscale. Rather than counting each individual bubble, it averages the amount of white and gray and black seen within the heart chambers. And that seems like it might be a better tool than counting bubbles alone, because as we all know, grayscale and counting bubbles can be a painful process. Session 3 was focused more on the systemic treatment of HHT. And Jim Gossage actually led us on a tour de force review of the various systemic therapies for HHT, concluding that, sort of good nasal hygiene and iron should be the foundation of treatment. Augmenting that with laser therapy, sclerotherapy, doxycycline, tranexamic acid, perhaps bevacizumab, pazopanib, and even thalidomide as a last resort. There was a lot of variability in what people use, but some agreements that all of these therapies have at least some potential for help, but we as clinicians have to minimize the side effects, and there is hope that some future medications will provide better outcomes. Other talks about systemic therapy included a Avastin experience. One of the take-home points there was one dose does not seem to fit every patient, right? There's a lot of variability in what patients need for maintenance, and that we might all have to personalize our approach for each patient. Another interesting thing I found was itraconazole, which is an antifungal medicine used for example, ringworm infections, has some degree of anti-VEGF activity, and it does seem to be effective at reducing the EPISTAXIS severity scores, but it did not actually have any effect on the hemoglobin levels, the amount of blood you have in your bloodstream, but it did seem to help with nosebleeds. And then everyone had a lot of excitement about the pomalidomide trial, which is in fact will be the first ever NIH-sponsored clinical trial in HHT patients. So we are looking forward to that. Session 5 talked about local treatments for HHT-associated pulmonary AVMs, and Justin McWilliams really tackled a lot of tough questions here with some near meta-analysis rigor. We were impressed with his talk, and the basic questions he focused on revolved around the following. Can we ignore small shunting that we see on contrast echocardiograms? So some patients have just a few bubbles here and there, and his conclusion was most likely yes. This is not a definitive, but a most likely yes, we can ignore small amounts of shunting. Should we treat all PAVMs? In particular, should we treat small pulmonary AVMs? His conclusion was probably, but the evidence for this was weak. Should we treat pulmonary AVMs in pregnant women? And the answer to this, his conclusion was most likely yes, we should do that. Should we treat PAVMs in children was sort of another big area of focus. And the conclusion there is, well, it depends on who you ask. It varies from center to center. It varies from continent to continent. And it was a topic of later discussion and actually continued discussion throughout the meeting. Cusimano actually discussed the challenges of retreating recanalized PAVMs. Distal occlusion is better. Distal is better for a longer occlusion, but it is technically difficult to perform. And then Schneider prospectively assessed cerebral complications of having a pulmonary AVM embolized in 94 patients, and there was only one patient who had evidence of a possible complication. It was a clinically silent embolization, but it's not sure if that was even related to the procedure. So the conclusion is that the procedure is safe. Session 5 focused on the local treatment of HHT epistaxis. A couple of interesting points. Tacrolimus or tacrolimus, depending on your preferred pronunciation scheme, seemed to be effective at mildly reducing but significantly reducing the epistaxis duration. And etamsulate spray did also reduce bleeding. Surgical therapies, including laser and sclerotherapy, also helped both quality of life and epistaxis severity score. And I think that's no surprise to most of us. Then we had a workshop focused on mechanism-based therapeutics. So you just heard from our colleagues a few minutes ago that BMP9 and 10 are involved in the mechanism, and in fact there are ligand-based therapies of BMP9 and BMP10 that are on the horizon and look promising. They'll probably come out first for pulmonary arterial hypertension treatment, and we're starting to get some sense of the side effects such as calcification and heart and liver toxicity, but there's at least some promising newer therapies for HHT. Tranexamic acid use was discussed with no clear answers for how to, what I would say is, navigate the complex scenarios in which you have to keep people's blood relatively thin, but you also are worried about clotting. So we have to balance bleeding and clotting, and no clear answers, but a lot of people had opinions about that. And finally, there were many people in the audience who used bevacizumab for epistaxis, but none of them really as a first-line therapy. We did point out that many patients have already had first-line prior therapy for this, and that was an interesting point. Okay. Our keynote speaker was Dr. Andrei Kinzelski from the NIH. He told some interesting stories about when he actually had to go to Chernobyl and his shoes became radioactive. He reiterated to us that there is in fact no magic bullet to get funding from the NIH, and he did talk about BioLink, which is an underutilized tissue repository, and encouraged us to focus on some studies that might use BioLink. Session 8 was talking about emerging issues in HHT. Marianne talked about the HHT Tracker app. Raj talked about some specific quality of life tools. We talked about a topical propranolol gel that has anti-angiogenic effects, and it seems to work without causing bradycardia. Interesting work on the SMAD4 pathways led to a recommendation that we screen patients every couple of years if you have a SMAD4 known mutation for malignancy. and to be aware of having problems with the aorta itself. And I think these were two areas that we as a community are not as focused on as we should be. The Yale group presented patients, 176 pediatric patients, and they noted that if your SATs were above 93%, there were no significant adverse events in this patient population, which is an interesting finding. Below 93%, there were some significant adverse effects. All right, and then nasal splints tend to be well tolerated, and people think they work. The workshop on pediatric pulmonary AVMs did kind of talk about the Yale study, but the ongoing two schools of thought, what happens kind of in some US centers where we screen for pulmonary AVMs with contrast echo, as opposed to some of the European centers where they screen with a pulse oximeter alone or a pulse oximeter and a chest x-ray. There was no agreement other than saying that screening should be done for children to make sure they don't have pulmonary AVMs. The method of screening is still somewhat controversial. That's safe to say. Although more will come out about that, we hope, with the new revised guidelines. There was a workshop on EPISTAXIS, and I think my take-home message here is there are a lot of great options, but a lot of variability in practice. Some things work for specific patients. Some tools work better in different physicians' hands. Local therapy with tacrolimus and propranolol, systemic therapy with bevacizumab and pazopanib, surgical management, sclerotherapy, and even sort of adjunctive therapy, right? If people have chronic allergies, maybe managing their allergic rhinitis with steroids may improve the amount of epistaxis, and that does seem to be the point. Genotype-phenotype correlations in session 11 talked about how, you know, we need to have more realistic goals, perhaps. We realize we can reduce the number of vascular anomalies, and we all like to have high hopes for cure, but hopefully with precision medicine and better understanding therapeutics in the future, we will be able to have some cures. Jamie McDonald talked about capillary malformation AVM syndrome and some of the overlap with HHT, as well as a new mutation. And then Hani Al-Samkari is leading the INHIBIT studies. Again, lots of variation among providers using bevacizumab for high output cardiac failure. Part of the issues are getting it authorized by insurance, but most people have not had difficulty with that. Session 12 was about brain AVMs. It was interesting, they were focused on non-HHT brain AVMs. A number of them are caused by KRAS mutations. Low risk features, in other words, unruptured, seem to be similar to HHT, so about 1% risk per AVM per year. Some conclusions that flow through the vascular malformation is important for the vessel malformation's growth. And so maybe we should be using other methods to measure the amount of flow through these. And then an interesting talk about COX-2 expression. So cyclooxygenase inhibitors might actually have a role in vessel inflammation. So medicines such as celecoxib need to be looked at and investigated to see if they can help. Seems like a number of take-home points, I think, from the clinical standpoint is we are getting better at making diagnoses, and in our investigations and our tools for figuring out how the organs are doing. We have systemic therapy, we have local therapy, and we have some interesting HHT trackers and other apps that are coming out that were useful. I think it was a nice clinical meeting. Finally, we wanted to thank Dr. Rojas for hosting, and Puerto Rico for hosting the meeting. It was a beautiful facility. It was a great sense of camaraderie and working together, and we were very happy to be there. And this is the final slide. There's a picture of yours truly and Dr. Marambaud, and we are happy to, I think, open it up for questions. Is that right, Nicole?
Yes, that is correct. So let me get Dr. Marambaud off mute. Okay. And if you all want to activate your webcam so everybody can see who you are. So thank you very much, both of you, for an incredibly thorough discussion of the basic science and the clinical science that went on during that almost 4-day conference. And so we'll just get started here. I think the first question is to Dr. Marambaud, and it's how are these breakthroughs in BMP10 related to future therapies, and what types of therapies or what type of research would be needed to advance those breakthroughs?
Yes, that's a very interesting question. The notion was that BMP9 is the most important ligand for ALK1 and endoglin, which are the main targets for HHT. And so recent research seems to indicate that BMP10 actually is also an important ligand, and in the context of HHT might be actually more important than BMP9. So basic research will be required to really make sure this is actually what's happening, that BMP10 is preponderantly important in the mechanism of control of ALK1 endoglin signaling. And as Dr. White mentioned, there is a lot of effort in trying to understand whether BMP9 infusion could be beneficial in the context of pulmonary arterial hypertension treatment. So an approach for HHT could be to try BMP9 as well, but also to switch to BMP10 and see how first in experimental model this is beneficial, and maybe later on to initiate a clinical trial.
Sounds great. Dr. White, did you have anything to add to that?
I agree.
Okay. So another sort of related question is just what are examples of BMP10, and would that be the same for ALK1 and endoglin patients?
So can you say again, what are the examples of BMP10?
Yes, and will it be the same for ALK1 and endoglin patients?
Oh, I see. So in the context of HHT, we believe that endoglin and ALK1 work together. They can form independent complexes. We call that complexes with other receptors. But in the context of HHT, it seems that they interact and bind BMP9 and BMP10. Whether 10 is more important than 9 is really context specific, and also due to the specific tissue that is expressing these receptors. So it's really an open question at the moment. Yeah.
Right. I agree. Yes, it's premature to start going to the drugstore and looking for these medications. They don't exist yet, and so we're early phase of trials at looking at them. And it's not clear which will work better, but there's some promise that at least that mechanism may be useful for HHT patients.
Great, thank you both. So the next question is, in lay terms, what would each of you say are the top 3 findings that would have a positive effect on HHT patients that you walked away with from the conference?
Andy, you want to start?
Maybe we do one and then one and then one. The top 3 findings would have an impact on. Well, I think that one that we're talking about right now is that there are some medications in the pipelines that have both a mechanistic rationale for use, like the BMP medicines, and some evidence that experimentally they may be useful. So right? Think back 10, 20 years ago when we did not have bevacizumab, right? These anti-angiogenics brought up a whole new, and we're still studying them, I admit, but a whole new sort of mechanistic tool that we can use to treat HHT. This is an entirely different sort of mechanistic tool that could be very useful. So that's my number one. I'm going to see if Philippe can come up with his number one. Then I'm going to come back and then we'll go back and forth.
Okay. I think my number one would be the same, really. A better understanding of the mechanism is allowing us to explore drugs that are already FDA approved, and that will really fast track some clinical trials, obviously, assuming, of course, that they are not causing too many side effects, especially in the context of a already fragile patient as an HHT patient. So, but that would be also my number one. Drug repurposing for HHT based on very good rationale coming from a better understanding of the mechanism and targeted treatments at the molecular level.
All right, my number two take home that was interesting has actually been the same sort of topic people have been talking about for a while, which is how much intervention is the right amount, right? Do we, and it was discussed and not necessarily answered to everybody's satisfaction, right? Do you embolize every single AVM that you can find? You know, there are, like we said, there are people who want to get rid of every AVM, and there are people who are happy watching them grow, taking, you know, a careful look for side effects. And so, you know, I think some of the take-home lessons from this particular meeting for me were that not everybody embolizes everything, and or treats everything necessarily, and judicious use of evaluation and investigation and therapy is probably the right way to go. Now, that's not saying you ignore symptoms, but it's also not saying you overreact. So you have to have a sort of a right balance for how you approach patients. That, to me, is number two.
So number two for me would be at the basic science level, would be a better understanding of what are the extracellular ligands that are defective in HHT, and whether we can really neutralize them if they are overexpressed and contribute to the formation of the lesion. This opens really a very great opportunity for immunotherapy, for instance. We talked about angiopoietin 2, which is elevated in several models, mouse models of HHT. There is an antibody that is currently in clinical trials for cancer, because this target, angiopoietin 2, is elevated also in some forms of cancer. So I think this is trying to go after what is accessible outside the cells, in the blood circulation, maybe with an antibody to develop immunotherapy approaches. That would be my number two. There is evidence now that maybe this is an interesting avenue.
All right, my third one, and I'll make it quick, is I thought I had this all worked out in my head. Is the realization that there are other things that happen besides nosebleeds, pulmonary AVMs, and you know, brain AVMs, such as the hepatic encephalopathy that may occur, and so we need to keep you know, keep an eye out for ammonia levels, the subaortic membranes that can occur, the possible screening of all SMAD4 patients every couple of years rather than every several years. So having a sort of a broader understanding of the other clinical symptoms and syndromes that occur. Back to you, Philippe.
And my number three will be probably the clear realization that there are second hits to cause the disease. This was speculated a long time ago. Some patients from the same family have very different phenotypes, very severe diseases in one sibling and not in the other. So clearly they are, in addition to the mutation we know in endoglin and ALK1 mostly, there are other hits. One of them could be allergy. Andy mentioned patients suffering from nasal allergies, and this is causing increased epistaxis incidence. There is this notion that maybe additional hit at the genetic level with this somatic mutation could be an important second hit. Inflammation could be also very related to that. And so more we understand about this second hit, more we open the box to more opportunities to treat them and not necessarily go after the genetic defects on endoglin and ALK1, but deal with the secondary hits that might prevent, in some ways, disease development.
Great. Yes, I'm going to remove one of my top threes and I'm going to replace that with that. I think that was a very important and convincing article. If, we all know that you accumulate vascular malformations over time, like on the fingertips, the lips, and the tongue. I have children, patients that have very few, if any, but we all know you get more with time. And so so if we can figure out a way to prevent that from happening, we may bypass some of the original troubles. Good.
Yeah, and clearly aging is
thank you.
some process in that situation. Yeah.
Right.
Very good. Thank you so much. So the next question we have is directed to Dr. Marambaud, and it is, how would you relate tacrolimus versus sirolimus regarding tolerability and dosing? Like, are they comparable medications?
So that would be more.
And is there any clinical trials in those areas?
Okay, so this would be more a question for Andy and with more experience in the clinical aspect of treating patients with these drugs. What we see at the experimental level is that sirolimus seems to be much more potent at equal amount in animal models in not only preventing but also reversing AVMs. My understanding is that they are both used for prophylaxis of solid organ rejection, and they have strong anti-inflammatory properties, so they are not easy to handle molecules. However, in the context of vascular malformation diseases, including in children, sirolimus is prescribed every day, and in some situations can be handled very easily. Andy, you want to add something?
I agree. I think they're equivalent medicines. They have different half-lives, but they're both used for transplant organ rejection, to prevent rejection. It just so happens that they also are useful here in blood vessel growth and minimizing inflammation. I'm not aware of any direct clinical trials, but on the screen, if you go to curehht.org, they have a connection for research and some ongoing projects, and so we can look there and see if there is anything going on. But they are useful for a number of different illnesses and diseases, not just HHT.
Great, thank you. So the next question is asking specifically about Avastin, also known as bevacizumab, and really just wanting to know, is there any research saying how long a person can be on it? For instance, this individual's been 2 years.
There have been people on it for years, but there's not the ideal research project that we would like, which is you take somebody who's been on it for 50 years and compare them to somebody who's been on it for 40 years and compare them to somebody who's been on it for 30 years, all while comparing them to somebody who hasn't been on it, has been on a placebo. So we don't have very long-term data about that. we do believe that we understand some of the side effects and that it is relatively safe to be on for years. We don't have that data in infants or children, but in adults we do.
Okay. The next question I think relates to Dr. Oliger's talk on liver AVMs and asking if family history showed any influence in screening recommendations for liver AVMs, particularly children whose parents have had significant liver complications.
Right, and that's been that's a topic of interest for many years, right? So some genetic, some particular mutations in HHT give you a slightly higher chance of having hepatic involvement than other mutations. But because of the variable penetrance in certain family members, we are not convinced that even if a parent has a bad family history of liver AVMs, for example, that any of their children or relatives will have the same degree of hepatic problems. That being said, we don't tend to look in children for liver AVMs because we don't yet have a good therapeutic intervention. That's number one. Number two, we don't see a lot of problems from the liver AVMs in children, even when we do look for them. So they're small, they don't shunt, they don't cause high output cardiac failure like you'll see in the adults. So the answer to the question is because of variable penetrance within family members, we are curious, but we don't necessarily look for everybody in the family at the, certainly in children, with risk for hepatic vascular malformations, limited because of our poor realm of interventions. I think I answered that right. Philippe, any comments?
No, I agree.
Okay.
Okay. Were any new studies discussed, or Andy, you mentioned the guidelines regarding asymptomatic and small brain AVMs, specifically in pediatric patients?
There were no studies about that at this meeting. I have one going on myself now, so I'm accumulating data as we speak.
Okay. And so again, I guess with the liver AVMs, just asking, based on what was discussed at the conference, what would be the suggested screening for liver AVMs?
We could not make any conclusions based on the discussion at the meetings, but I believe they're being addressed by the guidelines committee, and we look forward to hearing what those are going to be.
Okay. And our last question for the evening, I'll pose to both of you. What role would you see Cure HHT having to propel any of this research forward? And that's actually, I did not plant that question. It was asked to me.
Well, it would be nice, for example, if the Cure HHT Foundation could partner with some other organizations, for example, the Chan Zuckerberg Initiative, to help fund ongoing research and studies so that we can more definitively answer the questions that come to us and we're left scratching our heads sometimes.
Wow, you read our emails.
Just came out.
Yes, we're super excited about it.
Yeah.
Dr. Marambaud, how do you see the foundation helping move research forward?
So I think webinars like this one tonight are very helpful, not only to talk about basic research, but also try to connect the two and really start the discussion, and also hear from the patients and what are the priorities, and so really help us to connect better. Especially I'm talking about basic scientists who tend to be clustered in their labs and not seeing patients. This is helping tremendously, and this is a great thing.
Yes, and in fact, I'll echo
Fantastic.
that. You know, we had, at this meeting, we had an extraordinary group of individuals from across the world, all focused on one disease, but from many different aspects, right? We had clinicians, we had basic scientists, people focusing on blood vessels, we had people focusing on imaging. So getting people together to continue to work on our problems, I think is a great aspect of the foundation.
Yeah.
Fantastic. Well, thank you both very much for a very comprehensive overview of the basic science and clinical science, and helping tie it all together and what it means to the HHT community. And as Dr. White alluded to, we have recently been putting out an announcement that Cure HHT is one of 30 grantees for the Chan Zuckerberg Initiative, and we will be working towards setting up research networks and advancing research for HHT specifically, and there will be more information coming, but you can check our website for information and check your email for that announcement. I also want to remind everybody that the executive summary of the 2019 scientific conference, along with all of the abstracts that were submitted at that meeting, have been published in the professional journal Angiogenesis, and these articles are in the Cure HHT resource library. I will be providing a link in the email you'll receive after the webinar if you'd like to review those directly. Also, just a reminder that diseases can only be cured when scientists understand them, and this understanding, which comes from basic research, leads to new drugs and treatments. The evaluation of new drugs and treatments or the creation of new assessment tools require patient participation, and without you, the advancement of a cure for HHT is impossible. So please consider participating in HHT research as clinical trials are announced, or your input is requested through surveys or many other ways. On behalf of everyone at Cure HHT, we thank you for attending tonight's webinar. Our shared goal is to advance one common hope: to give those affected by HHT a chance for a normal life. This webinar series is just one of many ways we accomplish our mission, and we're grateful to the donors of Cure HHT for making this and all webinars possible. Please consider donating to Cure HHT to ensure we can continue to offer high-quality education for you and your families. And after the webinar, you will receive a short survey. Please take a few minutes to complete it so we can better serve you in the future. Again, I want to thank Dr. Marambaud and Dr. White for volunteering their time tonight to lead this webinar, which has been recorded and will be posted on our website at www.curehht.org in the resource library. We truly appreciate your participation and hope you enjoy the rest of your evening. Good night.
Good night.